everyone, I am Dr. Sonal Jain. In this series of forensic science lecture, today we are discussing about various DNA sequencing techniques used in various wildlife samples or other samples which we collect from the crime scene. So, the one technique is DNA nucleotide sequencing. The sequencing identifies each nucleotide base within a specific target region of DNA, the genetic marker, and the species identification usually involves sequencing around 500 bases of DNA to provide a species-specific sequence. These sequencing provides data for developing genetic markers such as single nucleotide polymorphisms, which is known as SNPs, microsatellites, which describe specific areas of sequence variation. So, SNP typing, when we talk about, this is another way we can sequence our DNA. SNP typing is single nucleotide polymorphism typing or genotyping, which allows specific variable sites in a DNA sequence to be investigated. Focusing on a SNP site allows faster, cheaper tests to be developed that do not require such long fragments of high quality DNA. So, basically the positive point with the SNP typing is that if you have a very less quantity of samples, that can be SNP genotyped. You do not require the long chain of DNA for this. So, as we know, the crime scene samples, they are very limited in quantity. Obviously, you will not get the large amount of samples at the crime scene. So, even if you have procured a single piece of sample, that can be sequenced using SNP genotyping. However, less information is gained in comparison to DNA sequencing, because in normal DNA sequencing, for example, if we talk about the capillary DNA sequencing, we get a big sequence, but in SNP DNA typing, we get the less information. There are multiple methods for typing SNP markers. For examples used in DNA forensics, one is PCR RFLP. So, PCR RFLP is which relies on the ability of enzymes to cut DNA at specific recognition sites. So, these enzymes, they have particular sequence and they cut DNA at various sites. Where these sites coincide with the SNP marker, some sequences will be cut to produce 2 DNA fragments and others will not, which lead to differences in the number as well as the length of the DNA fragments. So, how then we can identify that? We can identify further through the electrophoresis. So, then whatever the bands of DNA will come according to their size, the larger bands and the smaller bands, how the restriction enzyme has cut at the different sites, then we can observe that. Then we have allele specific PCR. So, in this PCR, the primers used in PCR for amplifying genetic markers can be designed for conserved regions of DNA, which is universal primers or regions where DNA varies between species or populations, which is known as the allele specific primers. The allele specific primers are designed so that PCR only works when DNA from the target sequence is present in a sample. So, allele specific probes are also there, which are different from the allele specific primers. How they are different from the primers? This is an alternative which is being used for the specific primers to use universal primers in combination with the specific probe. So, in this case we use different probes which are designed to attach to different DNA sequence variants, and they allow the base parent present at the SNP to be detected. Another example of the genotyping is microsatellite genotyping. In this, we use the microsatellite markers which describe differences between DNA sequences due to variation in the number of repetitive units of DNA in a specific region. Changes in the repeat number lead to different sized DNA fragments, and again we can use the same technique of electrophoresis to know that. Now, the genotyping is the stage where we are extracting the DNA and going for it. But there are another methods also through which can we directly identify any species and which are cheaper compared to the DNA extraction and other techniques. For example, the blood typing. We know that the blood proteins and enzymes have the characteristic of being polymorphisms or isoenzymes, which means they exist in several forms and variants. So, each one of them have different subtypes. For example, we have like PGM12, we have ADA, we have adenyl kinase. So, depending upon the different enzymes which are present, like transferrin is there, G6PD is there, 6-PGD is there, depending upon all these enzymes, we can get to know which species is present over there. So, each of these protein and enzyme variants as well as the blood types, they have a known distributions in a population. It is therefore a simple matter to calculate probability estimates that border on individualized blood typing. So, supposedly if you do the math, suppose you had a crime scene sample and a suspect which both were characterized by a type A blood and the basic subtype is A2. So, type A blood will give you that the 42% have type A blood, but in the type A blood having the subtype A2 you narrow down and comes to 25%. Then you look for the protein AK which is present in only 15% of the population. Then you again narrow it down to the 15% and the enzyme PGM2 you again narrow it down to the 6%. So, the probability of finding 2 people in the population with this exact type should be very less. That is, if we take the multiplication of these probability, that is 0.42 into 0.25 into 0.15 into 0.6, that gives you a probability of that particular species 0.000945. So, coming to the level, if we are even using the blood typing or the enzyme typing, you can come to a level where you do not have to go for the various DNA extraction techniques or the DNA analysis technique, and then you can further analyze using the enzyme typing or the blood typing. So, although I would say that DNA analysis has proved to be a reliable and sensitive tool for wildlife forensic investigations, enabling accurate species identification and individualization from biological traces, but the genetic results support the effective detection of illegal wildlife trade and poaching related evidences even from the degraded samples. So, now with the availability of PCRs and other techniques, we can use these techniques similarly for that. Another important studies which can be done on these wildlife samples are the stable isotope studies. So, what are the stable atoms or the unstable atoms? The stable atoms are the stable isotopes of the carbon 12 and the carbon 13, but the unstable isotope is carbon 14. These can be used to know about what these species ate and in the areas where they used to live. That means they comment on the paleo environment as well as the paleo diet of the species which we are examining. The stable isotope studies are very helpful in various forensic cases. For example, if a person is living in an area, let us talk about the human samples. If we are living in a particular area and we are staying there from the last 20, 25, 30 years and we are drinking water from that particular place, then the water content having all the minerals that will be slowly starting depositing in our bones as well as our hairs or our nails. So, when we analyze our hair samples, our nail samples, a brief point of time, then we can identify that because of the water we used to drink, our hair has those traces of that particular organical minerals which are present at that particular area. A case in the USA Texas area was being solved by using the stable isotope studies. So, to introduce this, the isotopes are the atoms with the same number of protons but differing number of neutrons. Isotopes are different forms of a single element. For example, we have carbon 12, carbon 13 which are both isotopes of carbon, one with 6 neutrons and one with 7 neutrons, but they both have the 6 protons. For example, oxygen 16, oxygen 17 and 18 are both isotopes of oxygen, one with 8 neutrons, one with 9 neutrons and one with 10 neutrons, but both having the 8 protons. So, these isotopes, these indicate the past changes in environmental processes that cause differences in the relative proportions of an element's isotopes present in the particular sample. So, we have unstable isotopes and stable isotopes. Unstable isotopes, we know all that they spontaneously fall apart and they emit particles and energy which is known as radioactivity, which are being used for various nuclear fusion reactions. But here we are discussing about the stable isotopes. So, what is the positive thing about these stable isotopes? They remain as they are indefinitely. For example, if a person has taken some particular diet, that will show in their body. So, the term arises or the saying arises from there that we are what we eat. So, the particular carbon, the oxygen levels, they will be maintained in our body all along because these isotopes, they remain there indefinitely. So, what are unstable isotopes good for and what are stable isotopes good for? Unstable, as we say, they are used for dating because we know a predictable rate at which these isotopes decay. But for the stable isotope, different isotopes of the same element are taken up differently depending upon various physical and biological conditions. The different masses cause isotopes to behave differently in physical and chemical processes. For example, if we say H2O is much heavier than 1H216O and will be left behind during evaporation. That means the chemical is similar, it is H2O, but what kind of mass the isotope is carrying, that will make it different. So, while using the stable isotopes for the identification of various wildlife animals, there are certain principles which we have to work on. The elements occur in the earth system in multiple isotopes, which could be both stable as well as unstable. And the proportion of stable isotopes in the earth system are constant over the entire system, but vary within different parts of the system. So, the ratio of stable isotopes were set when the earth formed and with slight changes as things falls to the earth. So, as we can see that molecular weight of water is 18. So, it makes up around 99.8% of water, which evaporates very easily and precipitates less easily. But we have a different water with a molecular weight of 18 and 20, which is the 0.2% of water, and it is 11% heavier than our normal, which H16O water, and they evaporates less easily and precipitates more easily. So, the atomic masses, the difference in the atomic masses of the oxygen 16 and the oxygen 18 make the variation in the water molecules also. This helps us in the identification of various specimens, that what kind of stable isotope is present. So, third fundamental premise is that the lighter isotopes are more reactive than the heavier isotopes. The lighter isotopes are more likely to be lost by diffusion or evaporation, whereas in higher temperatures, lighter isotopes are preferentially lost, and the biological systems, they will differentiate between different isotopes, which we can further use for our studies. They use different isotope abundance, which are expressed as ratio simple minus ratio standard over the ratio standard into 1000%. So, using this equation, we can get to know the delta O or the delta H depending upon what kind of isotope we are talking about. Now, why we are taking a ratio? Why not the direct measurement? So, the answer to this question is the differences between two isotopes of one element is very small. To measure them individually with enough precision is difficult and impossible in certain cases. So, by comparing a sample ratio to a standard ratio, the difference between these two can be determined much more precisely. So, since natural variations in isotope ratios are small, we can use the delta notation and get to know about that particular differences in the sample. Here we can see that the delta x, we can use any element we are talking about. R is the heavy or light isotope ratio for element x, and units are parts per thousand or parts per milliliter depending upon what we are using. So, the way we are write, we say the delta O18 or delta S34. So, depending upon we have to do this stable isotope studies, we take their ratio and then we analyze it. So, the majorly found which we do the stable isotope studies are on carbon, nitrogen and oxygen. 16 oxygen is the norm as we have studied, that is present around 99.8%, but the 18th isotope of oxygen is present in ocean in very small amounts, that is only 0.2%. 16 as we said is lighter and the 18 is heavier. So, sea water and fresh water have different isotopic signatures. There, from this point we can clearly say, supposedly there is a fish species which we have to identify. We can easily identify whether this species belong to the seawater species or the freshwater species depending upon their ratio of the oxygen present in them. So, as we see the cycle that the evaporation happens and the rain happens and the vapor precipitation and then the further formation of the vapor, that the water is being transferred and then again transferred back to the sea. So, this cycle goes on and the ratio is being maintained. So, that is why we can use the ratio in such cases. where we have to see the ratio of the 16O DNA as well as the 18O DNA. For this, different standard parameters are being used. For example, VMSMOW, Vienna Standard Mean Ocean Water, where we have bunch of ocean water kept in Austria. We have PDB, that is PD belemnite, which is a fossil of belemnite from the PED formation in Canada and which is being used for carbon and oxygen. So, depending upon what kind of stable isotope studies we are doing, what kind of elements we are working on, we can use different standards. Like another standard is CDT, Canyon Diablotrolite, which is a meteorite fragment from meteor crater in Arizona, and it contains FeS mineral trolite S. Then air is the best for the atmospheric air and for the nitrogen. So, now what makes for a stable isotope system that shows large variation? The first one is the low atomic mass, the second one is relatively large mass differences between the stable isotopes, and the third is the element tend to form highly covalent bonds. To go with that, we have element has more than one oxidation states or forms bond with a variety of different elements, and the rare isotopes aren't in too low abundance to be measured accurately. So, therefore, we go for the isotope fractionation. The isotope of an element have same number and distribution of electrons, hence they undergo the same chemical and physical reaction. The differences in mass can, however, influence the rate or the extent of the chemical or physical reactions, or they lead to the partitioning of isotopes differently among the phases. So, isotope sorting can be done during chemical, physical or biological processes, and that is known as the fractionation. The fractionation is a reaction or process which selects for one of the stable isotopes of a particular element. So, if the process selects for the heavier isotope, the reaction product is heavy, and if the process selects for the lighter isotope, the reaction product is the lighter. So, depending upon what kind of isotope is being selected, the reaction product will vary. So, the fractionation is being done on the isotope effect, where we can have the lighter isotope or we can have the heavier isotope of that. So, this isotope effect causes the fractionation. The fractionation factor which we use is alpha. R is the ratio of heavy to light isotopes. Alpha or fractionation factor is the ratio between the reactant and the product, where we can get to know what is the ratio of the reactants which we have been taken and what is the ratio of the products which are being coming out of the equation. So, while measuring isotopes, while different isotopes of the same element exist in certain fractions corresponding to their natural abundance, which can be adjusted by the fractionation. So, here the fractionation we can measure, where Ra is the ratio of heavy to light isotope and alpha is the fractionation factor. So, we measure isotopes as a ratio of the isotope versus the standard material in per milliliter %. So, that is why we multiply it with the 1000. Now, when the results are being observed, if delta is more than 0, that means the sample is enriched in the heavy isotope related to a standard, and if delta is less than 0, then the sample is depleted in the heavy isotope related to the standard. So, the relation between alpha and delta, they can be known through equilibrium versus kinetic fractionation, where fractionation is a reaction, but one in which the free energy differences are on the order of 1000 times smaller than other type of chemical reactions. So, we also go for the equilibrium versus the kinetic fractionation. Just like other chemical reactions, we can describe the proportion of reactants and products as an equilibrium or as a kinetic function. So, the isotope fractionation, the degree of fractionation depends on the relative weights of the isotope. Commonly fractioned are hydrogen, carbon, nitrogen, oxygen and sulfur. Somewhat fractionated is Fe, iron, chlorine, and fractionation impossible is in Be, fluorine, sodium, aluminium, phosphorus. So, depending upon what kind of material is there and the fractionation is possible, we can use accordingly. Isotope fractionation during chemical processes is caused by exchange reaction of the type half C16O2 plus H218O, and the equilibrium is maintained for half C18O2 and H216O. That means the oxygen can vary from here to there, from the lighter to the heavier isotope. And at the equilibrium we have for the preceding reaction, we use molar concentrations and not activities because the activity coefficients can cancel out. So, if CO2 and H2O did not discriminate between 16O and 18O, then K would be equal to unity. That means at that pertinent point they cannot be differentiated. But at 25 degree the K is 1.0 for 1, 2. So, which implies that carbon dioxide slightly prefers 18O and water prefers 16O. So, this preference is small, but it is large enough to cause a change in the isotopic fractionation. So, why we are studying this isotopic fractionation? So that we can see the difference between different isotopes which are present over there. The heavy isotopes, they form a lower energy bond and it does not vibrate as violently, therefore it forms a stronger bond in the compound. Here we use the rule of Bisleyson. The heavy isotope goes preferentially into the compound with the strongest bond. So, how does this fractionation work during the various different physical processes? So, we know that mass differences also give rise to fractionation during physical processes, diffusion, evaporation, freezing, etc. Fractionation during physical process is a result of differences in the velocities of isotopic molecules of the same compound. So, let us suppose we consider molecule in a gas. All molecules will have the same average kinetic energy and which is a function of temperature. So, we know the kinetic energy goes by half mv square, where m is the mass of the molecules and v is the velocity. Velocity, as we say, we know in which the direction the molecules are moving. So, because the kinetic energy for heavy and light isotopes is the same, we can write the velocity in terms of the mass. VL by VH is equal to MH by ML underscore. So, in this case of if we have CO2 with different isotopes, for example, we have one CO2 with 12C and another CO2 or CO with 13C. Now, put it into fractionation, we can see regardless of the temperature, the velocity of 12C is 1.0117 times that of 13C, because the 12C is a lighter molecule and we have a heavier isotope in 13C, which is a comparatively heavier molecule. So, the velocity of 12C16O will be comparatively lighter and compared to the molecule 13C16O. So, the lighter molecules will diffuse faster and they evaporate faster too. For the stable isotope measurement, we use a setup. We have auto sampler where sample can be put directly, whether it is a bone sample or it could be a axial sample or any other sample through which you have to go for it. We can get the dietary signatures from that. A diet high in C3 plants will leave behind the low levels of 13C and the diet high in C4 plants will leave high level of 13C in the bone collagen. Also, a diet high in marine food products results in high level of 13C, 15N and 34S. So, depending upon that, the stable isotope results demonstrate clear and interpretable isotopic pattern relevant to forensic reconstruction. The observed isotope ratios provide meaningful discrimination of dietary or geographic inputs, supporting their utility in individual profiling and provenance assessment. This consistency, when we see, which is being observed within the samples and expected variation between the groups, they indicate analytical reliability. Overall, when we say the findings reinforce the stable isotope analysis as a valuable complementary tool in forensic investigations. Usually, when we talk about this, if we can identify and we know the amount of the stable isotope will remain same in certain samples, then using the power of the fractionation, we can get to know what kind of stable isotope is present in that particular person and what kind of stable isotope is present in another person. On the basis of that, as we can say that other minerals or other things which are having a stable isotope, they can be also taken care of while we are looking for the samples in different locations. So, as a group of animals or as a group of humans, on which kind of species or which kind of plants they are feeding on, we have to look for that what kind of carbon, hydrogen and oxygen they are eating. So, the structure which they are having and they are storing that in their particular eggshells or in their body. So, going by that law, we can get to know about the paleodietary of those particular species. The paleodietary can be known about from the carbon which is present over there. We can go for the clumped carbon analysis, which is delta 47, or we can go for the triple oxygen isotope analysis. Now, when we are talking about the triple oxygen isotope analysis, we have to go for the triple oxygen isotopes, which are 16, 17 and 18. So, their abundance in the environment of different isotopes is different. Supposedly, if you have come across a species which is having a heavier water, they are drinking heavier water since they are staying at a place, then that particular heavier water isotope will reflect in their body parts. Any body parts we are talking about, if the sample is quite new, then we can see their tissues, we can see their hairs, or we can see their feathers, or in fact in their feces also. But if the samples are quite old, then we have to look for certain specimens in which these signatures are preferably settled down. For example, the hard materials which we can use, they are our eggshells, they are our bones, they are our antlers. So, as we say that the species, if they are eating a C3 plant diet or any other species which is eating a C4 plant diet, that can be easily well known through their isotope analysis. Now, depending upon what kind of samples we are coming across, we have to look for, or we have to see how we can extract the samples, or we can extract this ratio out of it. So, basically when we are going for the stable isotope analysis, we are using a combination of different GCs, that means the gas chromatography and the mass spectrophotometry. The mass spectrophotometry, we have a different mass spectrophotometer, that is the isotopic ICMS, we say ICRMS, where we have isotopic ratio mass spectrophotometry. So, what happens when we are connecting the GC with the IRMS, then we can get to know what kind of signals or what kind of different isotopes are present in that particular sample. So, the GC is being used at different stages. The sample is being administered using the autosampler. For example, if we are taking an example of the ostrich eggshells, the ostrich eggshells, while we are using in the autosampler, we are taking those eggshells and we are crushing those. So, as I have said before, like if you are using the preliminary techniques, we do not lose our sample, but in the confirmatory techniques, we tend to lose our samples. So, the stable isotope, the best part is we can easily identify about the dietary and the vegetation through which the species can be known from where they belong to. But the negative part of this technique is that the sample which we are using will be destroyed. So, depending upon what kind of technique will give you results, you can use a combination of preliminary as well as the confirmatory technique, and that will give you the best results for your forensic analysis. Thank you so much.